Finish eating, push the plate away. Notice what you do next. Most people do one of two things. They sit at the table for a while, talking or scrolling, or they move directly to the couch, the desk, or the car. The meal is done. The decision has been made. What follows is rest. The body disagrees with this framing. From the body's perspective, the meal ending is not the end of the metabolic event. It is the beginning of the most consequential 20 minutes in the body's daily biochemical schedule. Here is what is happening in the bloodstream in the minutes after a typical meal. Glucose absorbed from the small intestine is entering the portal circulation and arriving at the liver. The liver is directing some of it to glycogen storage and releasing the rest into systemic circulation. Blood glucose is rising. In a healthy adult eating a moderate meal, the peak occurs approximately 45 to 60 minutes after the first bite and reaches somewhere between 120 and 140 milligrams per deciliter before insulin clears it back toward fasting levels. In the 88% of American adults who have some degree of metabolic dysfunction, the figure published by the National Institutes of Health's 2019 analysis of NANE's data, that peak is higher, occurs later, and takes longer to clear. During the glucose peak, three things are happening simultaneously that determine the rate at which biological aging proceeds. The first is glycation. Glucose in the bloodstream reacts non-enzymatically with proteins, a chemical process that does not require any enzyme and cannot be turned off, only slowed. The rate of this reaction is proportional to the glucose concentration. Higher blood glucose means faster glycation of hemoglobin, collagen, arterial wall proteins, lens proteins in the eye, and neuronal proteins in the brain. The glycated products, advanced glycation end products, or AGEs, accumulate in tissues over years and decades, cross-linking proteins, stiffening structures, and driving inflammatory signaling through receptors called RAGE that recognize the AGE molecules and mount an immune response to them. The second is oxidative stress in the arterial endothelium. When glucose enters endothelial cells lining the blood vessels, it drives a chain of mitochondrial reactions that produces reactive oxygen species, free radicals, at a rate proportional to the glucose load. The mitochondria of endothelial cells, flooded with glucose during the postprandial peak, generate oxidative stress that damages the endothelial surface, reduces nitric oxide production, and initiates the inflammatory cascade that leads to arterial plaque formation. The third is insulin's own signaling consequences. Insulin released to manage the glucose peak drives glucose into cells, which is its job. But in chronically insulin-resistant individuals, the insulin signal is blunted, and the pancreas must release more insulin to achieve the same glucose clearance. Elevated insulin itself signals fat storage, promotes inflammatory cytokine expression, and suppresses the fat-burning metabolic pathways that the body needs between meals. All three processes are happening in the 20 minutes after you finish eating. The chair you sit in during those 20 minutes is either making them better or making them worse. If you're enjoying this story so far, don't forget to hit the like button and subscribe. Drop a comment telling me where in the world you're watching from. I love hearing from you. The shape of the glucose curve after a meal is not fixed by the food. It is modified by what the muscles are doing while digestion proceeds. Skeletal muscle is the largest glucose disposal organ in the body. At rest, muscle accounts for approximately 30 to 40% of postprandial glucose uptake. During gentle movement, walking at a comfortable pace, muscle glucose uptake increases substantially, not because intense exercise is happening, but because the muscle contractions during walking activate GLUT4 glucose transporter translocation to the cell surface through a pathway that is independent of insulin. This is the critical detail. Normally, GLUT4 transporters, the proteins that move glucose from the bloodstream into muscle cells, sit inside the cell until insulin signals them to migrate to the cell surface. During exercise, including gentle walking, GLUT4 translocation occurs through a separate pathway driven by AMP-activated protein kinase, the cellular energy sensor that detects increased ATP consumption in contracting muscle and responds by opening glucose uptake channels, regardless of whether insulin is present. A 10-minute walk after a meal activates GLUT4 translocation in the large muscle groups of the legs, the quadriceps, hamstrings, calves, and gluteals, and increases glucose uptake in those muscles during exactly the window when blood glucose is peaking. The glucose that would otherwise remain in circulation, driving glycation and endothelial oxidative stress, is being pulled into muscle instead. DiPietro and colleagues at the George Washington University Milken Institute School of Public Health published a study in diabetes care in 2013 that directly compared the effects of a single 15-minute walk after each meal versus a single 45-minute walk at one time per day on 24-hour blood glucose in older adults at risk for type 2 diabetes. The 3 15-minute post-meal walks reduced the postprandial glucose excursion by 12% more than the single 45-minute walk. The same total time of walking produced better blood glucose outcomes when distributed across the post-meal windows than when concentrated into one session. The implication is counterintuitive but mechanistically precise. The timing of gentle movement matters more than the total volume of movement for postprandial glucose control. A 10-minute walk after dinner is doing something for blood glucose regulation that a 10-minute walk at 6 a.m. cannot replicate because the glucose is not in the bloodstream at 6 a.m. the way it is 20 minutes after dinner. Postprandial glucose control is one of the most sensitive biomarkers of biological aging pace. The time spent above 140 milligrams per deciliter, the glucose level at which endothelial glucose toxicity has been documented in research by Ceriello and colleagues at the University of Udine, is directly proportional to the rate of glycation and the arterial oxidative stress load. Each hour spent above that threshold is an hour of accelerated tissue cross-linking. Each meal followed by gentle movement is a meal that reduces the time above that threshold. The arterial endothelium, the single cell layer lining the inner surface of every blood vessel, is the most metabolically active tissue in the cardiovascular system, and the tissue most directly exposed to the glucose and insulin signals arriving after every meal. During the postprandial period, the endothelium faces a specific challenge. The glucose flooding into endothelial cells drives mitochondrial electron transport at a rate that exceeds the cell's antioxidant capacity, generating superoxide and other reactive oxygen species that inactivate nitric oxide. Nitric oxide, produced by endothelial nitric oxide synthase in the endothelial cell wall, is the primary vasodilator and anti-inflammatory molecule in the vascular system. When it is quenched by postprandial oxidative stress, the arteries become temporarily less compliant, blood pressure rises slightly, and the inflammatory signaling in the endothelium increases. This postprandial endothelial dysfunction was documented by Plotnick and colleagues at the University of Maryland in a paper published in JAMA in 1997, showing that a high-fat meal produced measurable impairment of brachial artery flow-mediated dilation, a clinical measure of endothelial function, for up to 3 to 4 hours after eating. The artery lost some of its ability to dilate in response to increased blood flow during the postprandial window. The endothelium was temporarily less functional because the meal had driven oxidative stress that was quenching its nitric oxide production. What reverses postprandial endothelial dysfunction is exactly what generates shear stress in the arterial wall. When blood flow increases during gentle walking, the increased velocity of blood moving through the arteries bends the glycocalyx, the carbohydrate coating on endothelial cells, and activates eNOS through a mechanosensitive pathway. The same enzyme whose activity was being suppressed by oxidative stress is being stimulated by the mechanical signal of increased blood flow. The shear stress during a brisk 10-minute walk produces eNOS activation in the femoral, iliac, and aortic endothelium that partially counteracts the postprandial oxidative stress. The nitric oxide production that the glucose spike was suppressing is being driven back upward by the mechanical signal of moving blood. The arterial compliance that the meal temporarily reduced is being partially restored by the walk that follows it. This is not a metaphor. Arterial pulse wave velocity, the clinical measure of arterial stiffness, has been measured before and after post-meal walks in multiple studies. Miyashita and colleagues at Waseda University in Japan published data in hypertension research in 2008 showing that a 10-minute walk immediately after a meal produced significantly lower arterial stiffness measurements 60 minutes post-meal compared to sitting after the same meal. The arteries were measurably more compliant after the walk than after sitting, despite identical meals in both conditions. Each day that includes post-meal movement is a day of lower average postprandial arterial stiffness. Each year of lower average postprandial arterial stiffness is a year of slower arterial wall remodeling toward the stiff, less compliant state that characterizes cardiovascular aging. The 20-minute window is not preventing individual meals from mattering. It is determining the cumulative vascular age that decades of those meals will produce. Insulin resistance is the most widespread metabolic disorder in the developed world. The underlying condition in type 2 diabetes, metabolic syndrome, non-alcoholic fatty liver disease, polycystic ovarian syndrome, and much of the cardiovascular risk that does not fit neatly into the cholesterol narrative. It is not primarily a pancreatic disorder. It is a failure of the tissue that should be receiving glucose, primarily skeletal muscle, to respond adequately to insulin's signal. The muscle's insulin sensitivity is not a fixed property. It fluctuates on a timescale of hours, influenced by the most recent bout of physical activity. After exercise, even gentle exercise, GLUT4 expression on the muscle cell surface remains elevated for several hours, improving the muscle's capacity to take up glucose in response to insulin during that window. The effect is local. The muscles that were active show improved insulin sensitivity. The muscles that were inactive do not. Holozzi and Narahara at Washington University published the foundational work on exercise-induced GLUT4 translocation in the 1980s, establishing the mechanism by which muscle contraction improves glucose uptake independently of insulin. Subsequent work by Goodyear and Kahn at Harvard's Joslin Diabetes Center mapped the specific signaling pathways, AMP and KMK2 activation during contraction, followed by downstream GLUT4 translocation to the cell surface, and established that the effect persists for 2 to 3 hours after a bout of exercise before returning to baseline. The timing implication is precise. The muscles that walk for 10 minutes after a meal are the muscles with elevated GLUT4 expression during the postprandial window when glucose is peaking. They are pulling glucose from the bloodstream at a higher rate than they would be pulling it if the person had not walked. The pancreas, detecting the more rapid glucose clearance, releases less insulin to achieve the same blood glucose level. Less insulin means less insulin-driven fat storage signaling, less insulin-driven inflammatory cytokine expression, and less demand on the pancreatic beta cells that must manufacture the insulin. The cells whose progressive exhaustion over years of chronic insulin demand is the mechanism of type 2 diabetes progression. Post-meal walking is doing something that a walk at any other time of day cannot do as effectively. It is activating the glucose uptake machinery in exactly the tissue that needs to receive the glucose that is currently peaking in the bloodstream, reducing the insulin demand, and giving the beta cells a lower load day than they would otherwise have. Repeated across every meal, across years, this difference in daily beta cell load is the difference between preserved insulin sensitivity in late middle age and its progressive deterioration. If you're hooked on this story, make sure to subscribe and turn on notifications so you never miss an upload. Your support means everything and helps me bring you more emotional stories like this one. The mitochondria in skeletal muscle are not static. Their efficiency, the ratio of ATP produced per unit of oxygen consumed and substrate burned, depends on the demands placed on them in the hours after meals. When a large glucose load arrives in the bloodstream and is taken up by resting muscle, the mitochondria receive more substrate than their current ATP demand requires. The electron transport chain, flooded with electrons from the NADH and FADH2 produced by glycolysis and the citric acid cycle, generates reactive oxygen species at an elevated rate when the rate of electron delivery exceeds the rate of ATP synthesis. This mitochondrial uncoupling under substrate excess, a phenomenon documented extensively by Brand and colleagues at the MRC Mitochondrial Biology Unit in Cambridge, is one of the primary mechanisms of cellular aging in post-mitotic tissues. The reactive oxygen species generated by substrate excess mitochondria damage mitochondrial DNA, oxidize mitochondrial membrane lipids, and impair the function of the electron transport chain proteins themselves, reducing their efficiency with each oxidative insult. Over years, the cumulative mitochondrial damage from repeated postprandial substrate overflow produces mitochondria that are less efficient, more leaky, and less capable of generating the ATP that cells require for their functions. Gentle movement after meals changes the substrate demand balance in skeletal muscle mitochondria. When the leg muscles are walking, their ATP demand is elevated. The same glucose that would have overwhelmed resting mitochondria's capacity to match substrate with demand is now being consumed by contracting muscle at a rate that keeps the electron transport chain loaded but not overloaded. The reactive oxygen species generation falls because the imbalance between substrate delivery and ATP demand is reduced. The mitochondria are running at a load that matches their capacity. Tanner and colleagues at the University of Utah published data in Diabetes in 2002 showing that mitochondrial function in skeletal muscle was significantly impaired in individuals with insulin resistance compared to insulin-sensitive controls, and that the impairment was partly explained by differences in the oxidative stress load the mitochondria experienced under high glucose conditions. The mitochondrial damage was downstream of the metabolic disorganization, the repeated episodes of substrate excess meeting resting mitochondria, that insulin resistance represents. The post-meal walk is not primarily a cardiovascular exercise. It is a mitochondrial protection strategy. It transforms the metabolic environment of the postprandial window from one in which substrate floods resting mitochondria and generates oxidative damage to one in which substrate meets working mitochondria and generates ATP efficiently. The difference, accumulated across every meal of every year, is the difference between mitochondria that age at the expected rate and mitochondria that age faster. The 20-minute post-meal window is not equally consequential after every meal. The evening meal, dinner, produces the most significant postprandial metabolic consequences, and the post-dinner window is where the difference between sitting and walking is largest. The reason is circadian biology. Insulin sensitivity follows a circadian pattern, peaking in the morning and declining through the day to its nadir in the evening hours. The same meal eaten at 7 a.m. and at 7 p.m. produces different postprandial glucose responses in the same person, with the evening meal producing a higher and more prolonged glucose elevation despite identical food content. Rutrakel and Van Cotter at the University of Chicago, whose research on circadian disruption and metabolic function has been published across decades of work, established that the evening decline in insulin sensitivity is driven by cortisol's rhythm, the evening rise in growth hormone, and the circadian clock-driven reduction in GLUT4 expression in peripheral tissues. The practical consequence is that the post-dinner window, typically the largest meal of the day occurring at the point of lowest daily insulin sensitivity, is the window of highest glycation rate, highest postprandial arterial oxidative stress, and highest risk of exceeding the 140 mg per deciliter threshold, where endothelial glucose toxicity has been documented. Sachin Panda's research at the Salk Institute on time-restricted eating and circadian metabolism, published in Cell Metabolism and related journals, showed that aligning food intake with the hours of highest insulin sensitivity, the morning and midday hours, produced better metabolic outcomes than the same food eaten in the evening, through exactly this circadian variation in glucose disposal capacity. The circadian clock in the pancreas, the liver, the muscle, and the endothelium are all set to handle glucose most efficiently in the morning. The dinner meal is arriving when that efficiency is at its lowest point. This is not an argument for skipping dinner. It is the mechanism that makes the post-dinner walk specifically valuable. It partially compensates for the evening reduction in insulin sensitivity by activating the insulin-independent GLUT4 translocation pathway that walking drives, reducing the postprandial peak that the circadian clock would otherwise allow to run higher. A 10-minute walk after dinner is not doing the same thing as a 10-minute walk after breakfast. It is compensating for a specific circadian deficit in glucose disposal capacity that makes the evening postprandial window the most metabolically costly window of the day. The post-meal movement protocol is simpler than everything it accomplishes suggests. 10 minutes of walking at a comfortable pace, not exercise intensity, not elevated heart rate, not sweating, began within 20 minutes of finishing a meal. The pace should allow easy conversation. The specific metric that matters is that the large muscles of the legs are contracting, activating AMP, driving GLUT4 translocation, and pulling glucose from the bloodstream during the window when it would otherwise be peaking. The sequence matters. The walk must begin within 20 minutes of finishing the meal, before the glucose peak has occurred and begun its clearance. A walk begun 90 minutes after dinner is a walk that starts after the glucose has already peaked, driven its glycation rate, produced its endothelial oxidative stress, and begun its insulin-driven clearance. The glucose peak has occurred. The window has closed. The walk at that point is beneficial for other reasons but cannot accomplish what the early post-meal walk accomplishes. Timing within the 20-minute window. Most people can begin walking within 5 to 10 minutes of finishing a meal without digestive discomfort. The food has not yet cleared the stomach. The process of gastric emptying takes 2 to 4 hours for a typical meal. So the walk is occurring while food is still being processed in the stomach and while glucose is beginning to absorb from the small intestine. The muscle activation is timed to match the glucose absorption curve. The duration is 10 minutes for most meals, with the post-dinner walk extended to 15 minutes when feasible given the circadian deficit in insulin sensitivity that makes the evening window more metabolically consequential. Outdoor walking is better than indoor treadmill walking for one reason that is specific to the post-meal window. Light exposure during the post-dinner walk in the late afternoon or early evening provides the retinal light stimulus that synchronizes the peripheral clocks in the liver, muscle, and pancreas through the suprachiasmatic nucleus and the downstream circadian signaling cascade. Evening light exposure at the wavelengths present in the outdoor sky an hour before sunset, predominantly longer wavelengths, low intensity, does not significantly suppress melatonin the way blue heavy evening light does, but provides a meaningful circadian entrainment signal that improves the timing accuracy of the peripheral metabolic clocks for the following day's glucose management. The 28-day outcome of consistent post-meal walking is measurable in continuous glucose monitoring data, which has become accessible to non-diabetic individuals through devices like the Freestyle Libre and Dexterity CGM systems. The average time spent above 140 milligrams per deciliter decreases. The postprandial peak values decrease. The time for glucose to return to baseline shortens. These are direct measurements of the reduction in daily glycation load and endothelial oxidative stress that the post-meal walks are producing. The one-year outcome is measurable in HbA1c, glycated hemoglobin, the clinical marker that reflects average glucose levels over the preceding 3 months by measuring how much of the hemoglobin in red blood cells has been glycated. In individuals without diabetes, HbA1c typically sits between 4.8 and 5.6%. Each 0.1% reduction in HbA1c within the normal range corresponds to a reduction in glycation occurring throughout all tissues, not just hemoglobin, proportional to the improvement in average glucose control. The 10-year outcome is measurable in arterial pulse wave velocity, the direct measure of arterial stiffness that predicts cardiovascular events more accurately than most traditional risk factors. The arteries of people who have spent 10 years generating lower postprandial glucose peaks and better endothelial shear stress through post-meal walking are measurably more compliant than the arteries of people who ate similar diets and sat after every meal, not by a dramatic margin. By the margin that accumulates from 10 years of daily decisions made in the 20 minutes after every meal, 3 times per day, 365 days per year, 10,950 times. The meal ends. The decision begins. It is not the most dramatic health decision you will make today. It is one of the most consequential ones, because it is made 3 times per day, and because its effect is to change the chemistry of the postprandial window, the daily window when more of the biochemical work of aging is happening than in any other 20-minute period of the day. 10 minutes of walking, 3 times per day, within 20 minutes of finishing the meal. Nothing about this requires equipment, a gym, a fitness identity, or a significant time commitment. It requires only the understanding of what the 20 minutes after eating actually are, not rest after effort, but the beginning of the body's most important daily metabolic work, and the window in which your participation makes the largest difference to how that work turns out. Thank you so much for watching until the end. If you loved this story, check out the other videos on your screen now. I think you'll really enjoy them. And don't forget to subscribe if you haven't already. See you in the next story.